All right, so we are on week 4. Today we are diving into photosynthesis and respiration. We touched on that a little bit a few weeks ago, but we're really going to take a deep dive into respiration this week because it has to do with your sourdough starter project. So let's get into it. So we know that photosynthesis is incredibly important. It's probably one of the most important biological reactions that occur. It's carried out by plants, algae, and bacteria, anything that has chloroplasts. Those are the main cells that carry out photosynthesis, and then chlorophyll pigment. So mainly chloroplasts and chlorophyll are found in plants, algae, and bacteria, whether they're terrestrial, so they live on land, or even marine algae. Microorganisms can carry out photosynthesis as well. Like we learned, photosynthesis takes carbon dioxide, which is CO2, and water, H2O, along with sunlight, and creates glucose, C6H12O6, and oxygen, O2. All right, so that's the glucose down there, and oxygen is the two products of photosynthesis. We also learned a few weeks ago that pretty much the exact opposite of photosynthesis is another reaction called cellular respiration. This is where it takes the sugars, glucose, and oxygen, turns it back into H2O and water, all right? Or H2O, sorry, is water, and then CO2, carbon dioxide. So we have photosynthesis, carbon dioxide, water, turns into oxygen and sugar. Cellular respiration takes sugar and oxygen, turns it into water and carbon dioxide. Pretty much the reverse of each other. Furthermore, cellular respiration also creates ATP, which is energy of the cell. And they do this through glycolysis. So glycolysis is the breakdown of glucose into energy and pyruvic acid, which is more readily available and used by cells. So glucose isn't really a great energy for cells, but the fact that it's turned into pyruvic acid through glycolysis, that allows cells to use that energy. After glycolysis occurs, there's two forms of cellular respiration that can occur. There's aerobic and anaerobic respiration. Aerobic respiration occurs in the presence of oxygen, and that itself creates 32 ATP molecules. But overall, the cellular respiration with oxygen, glycolysis all the way through, nets about 36 ATP molecules. So you have oxygen present, that is aerobic respiration. Now, if there's no oxygen present, there's no oxygen within the system, cells can undergo anaerobic respiration, also known as fermentation. And this is characterized by an incomplete breakdown of glucose and only yields about 2 ATP molecules per glucose. Much less efficient than aerobic respiration in the presence of oxygen. So I keep saying this stuff, ATP. Well, ATP is the energy currency of the cell. It's what really makes cells tick. This is where cells get all their energy from. It's called ATP, also known as adenosine triphosphate, but ATP for short. So here again, we have our cellular respiration equation where we take the C6H12O6, which is glucose, plus oxygen, turn it into H2O and CO2, plus 36 ATP molecules. And this is just a little schematic of how that is done. After glycolysis occurs, where there's oxygen present, or oxygen absent, excuse me, there's fermentation. Where there's oxygen present, it's aerobic respiration. Now, breaking down this a little bit further, in aerobic respiration, where there is oxygen present, pyruvate is broken down completely in something known as the Krebs cycle. This also releases energy. This is about 2 ATP. The energy from the Krebs cycle is then transferred to the electron transport system, or the ETS, where it is further broken down and creates H2O and 32 ATP. I'm going to show a schematic of this in 1 second, so it's going to make a little bit more sense. And all this, the Krebs cycle, the electron transport system, the breakdown of everything takes place in the mitochondria of the cell, also known as the powerhouse of the cell. So hopefully this is a little visual schematic here. Remember, this is aerobic respiration, so this is in the presence of oxygen. You have glycolysis, which breaks down glucose into pyruvate. This releases energy in itself and with a net energy of 2 ATP. Pyruvate is then further broken down through the Krebs cycle in the mitochondria. This itself releases another 2 ATP, and then furthermore, in the presence of oxygen, this oxygen gets transported to the electron transport system, and then the final chemical reaction that goes on there nets 32 ATP. So this is why the aerobic respiration in total nets 36 ATP. 2 plus 2 plus 32 is 36 ATP. Any questions on the aerobic respiration side of things? All right. Now we switch to anaerobic, where there's no oxygen present. Glucose is broken down via glycolysis, again, into 2 pyruvate molecules and 2 ATP molecules. However, there are harmful byproducts from this reaction here. In animal cells, pyruvate is converted into lactic acid, which is what you get when you have sore muscles. And in some microorganisms, pyruvate is converted into alcohol via fermentation. And this is what kind of sets the standard for your sourdough starter project. Through the process of fermentation, you can see that the microorganisms, the yeast, in the presence of no oxygen, is converting all the sugars found in the starches in the yeast into alcohol via fermentation. All right, so this represents a very inefficient breakdown of glucose. Where there is no oxygen present, the whole system does not break down glucose as well as it does when there is oxygen present. And therefore, there's only 2 ATP molecules produced per glucose molecule. So a lot less ATP when you've got no oxygen present. So this is what kind of that it looks like. So you have glucose, again, through glycolysis, that's broken down into pyruvate. If you have in yeast cells, other microorganisms, the yeast cells create alcohol through fermentation. And then in animal cells and some bacteria cells, the byproducts of fermentation of anaerobic respiration are lactic acid. So again, only 2 ATP energy cells are created through this process, and which is not efficient for us. So if we didn't have aerobic respiration, our metabolisms would completely shut down. So here, just to review this whole thing for the day, again, glucose turns into pyruvate through glycolysis. Without oxygen, you have anaerobic respiration with harmful byproducts such as alcohol and lactic acid, depending on if you're an animal or a microorganism. And this only nets 2 ATP, 2 energy molecules. And then through aerobic respiration with oxygen, you have the Krebs cycle, electron transport chain converted to 36 ATP within the mitochondria of the cell. I know a lot of stuff thrown at you here in a short amount of time, but is there any questions on this? It's pretty straightforward. Nothing too much into it. I'm not going to go into the details of the Krebs cycle and the electron transport chain, but is there any questions on this so far? I wanted to leave some time today to talk about next week because there is a few things up and coming. So if we have no questions on this, I will move on. So next week, you got the sourdough starter project due. Some of you already turned that into me this week. Totally fine if you did. It just means you're a week ahead of schedule. You can kind of take a week off, but officially the photo of your sourdough starter is due next week. So hopefully you've got your materials together. If you haven't started yet, hopefully you have those. You don't have to scramble to get those. You're going to follow the instructions in your lab manual. I think it's page, what page does this one start on? Page 23, or page 22 actually tells you how to start it. You'll use this platform, the Students Discover lesson at Sourdough Starter. This is where you'll have your data pretty much installed, and this is where the data platform is stored. So this experiment is going to run for 14 days, but it requires attention every day. So make sure you're entering that data uploaded to the website every single day. It's stuff like, how tall is this concoction getting? What does it smell like? Is there any changes? So please note that in on the website there. So like I said, some of you guys have already turned this in. Just go ahead and start it off. Start your 14 years. This would be week 2 for you guys then. Last week was week 1. For those of you who did not turn it in yet, not a problem. It'll be week 1 this week, and then you'll turn in the selfie to me next week. So depending on where you are, if you're ahead of the game, that's cool. If not, don't worry about it. It's not officially due until next week. For those of you who have started on the sourdough, were there any issues, any questions? Did you guys get this far to registering on the website? Anything you want to share, maybe to help some people that maybe didn't start it? Any information would be helpful for those people. But it's pretty self-explanatory. Follow along in your lab manual. Anything else, if you have any other questions, just please always email me. Anyone want to shout anything out that perhaps did it? All right, no worries. So again, due next week by 11 a.m. is your sourdough starter. Send pictures of your sourdough for points. Not due next week, but you should be working on your ecosphere weekly measurements, thinking about how you want to manipulate it, and then taking daily measurements of your sourdough starter. You don't have to send me the daily measurements, but you do need to send me just a photo of you starting it. Again, no need to send me data sheets of your ecosphere. Just make sure you're keeping those for your final workup in a few weeks here. Hopefully everyone's ecosphere is still alive and thriving, but so start thinking about how you can manipulate it and turn it into an experiment. I'm going to do a little different thing on late assignments. So I've been pretty relaxed on all late assignments thus far. I mean, it's been, working with online classes. This is my first time working with Canvas, doing Zoom. So I let a lot of things slide, but I think we've all pretty much established ourselves online here. The deadline to drop is passed, so everyone should be locked into the class. So with that, the late assignments will now incur a penalty. So again, all assignments are due 11 a.m. on your class day, which is Wednesday. If you don't turn it in before 11 a.m., you'll be a 10% penalty for every day that it's late, up to 2 days, after which I will no longer accept the assignment. So if you turn in your sourdough picture next week after 11 a.m., the maximum points you can receive are 18 out of 20. If you turn in the next day on Thursday, the maximum points is 16 out of 20. And then if you turn in on Friday, maximum points you can get is 14 out of 20. So that's not a new late assignment policy. I think for this class, everyone's pretty on top of it, so I don't see too many people losing points, but I just wanted to throw that out there, let you guys know. If you notice on your schedule, there's also an exam next week. This will be done on Canvas. It'll be done at noon. So after the lecture next week, I've set it to exam start at noon, and you'll have 30 minutes to complete the exam. It's going to be roughly about 30 questions, maybe a little bit less, but roughly 30 questions. And the exam is open note and open book, but not open internet. So Canvas can track those things. So please just, have your notes in front of you, have your book in front of you, but don't be clicking through the internet to find answers to the questions. It's going to be mostly multiple choice, some true, false, some fill in the blank, but for the majority, it's going to be all multiple choice. And then everything that we've covered thus far in class and everything in the lab manual is fair game for the exam. I'm not going to quiz you. You got a question here. Can you have the PowerPoints open? No, I don't think so, because that would be open internet. Yeah, there's a way that Canvas checks whether you click off the screen and whatnot. So I would just have your notes in front of you, take notes off the PowerPoints beforehand. Yeah, so everything in the lab manual is fair game, as well as my PowerPoints. I'm not going to quiz you on stuff like, let's see, what's an example? not going to quiz you on like any of the procedures from the ecosphere or stuff like that. I'm not going to be like, oh, how much, you know, substrate should be using in your ecosphere. That's not stuff that's going to be on the exam. It's going to be more of the conceptual stuff that we learned, sort of the background information that we've learned in the class thus far. Any questions on the exam? Any questions on the exam? Again, 30 minutes to complete. If you need extra time, you know, I'm going to give you that extra time. If you have one of those student success things, you know, please turn that into me sooner rather than later. I know some people have already, but everyone, if you don't have one of those, it's going to be 30 minutes long, roughly 30 questions. And that'll be available noon after lecture next week. Any questions on today? All right, I'll stick around for people that have questions, but for the most part, you guys are good to go.